Okay, so let's talk about the structure of DNA and as well as DNA replication. So I'm going to be very brief when we talk about the structure of DNA. So our understanding of DNA started as we know it really is less than 100 years old, right? We knew something was in our bodies. We knew there was something that we were getting from our parents. We didn't know what that was. So in the 1950s, you had Watson and Crick, who were the ones that were credited with, of course, discovering or really uncovering, I should say, the structure of DNA and the double helix, and everything's great. You know, our long national nightmare is over. We now know what DNA looks like. So there's a ton of history here, which I'm not going to go into. I'm going to focus on two main parts of it. So DNA, what it is, it's deoxy, right? So deoxy, so one less oxygen, ribose, sugar, nucleic acid. So the sugar part is here, right? That's ribose. But it's minus one oxygen. Deoxy, minus an oxygen. You also have your phosphate group, and then you have your nucleic acid. And the nucleic acid are those bases. Adenine, guanine, cytosine, cytosine, thymine, those are your bases, okay? So you've got your base, which changes, your sugar, your ribose minus an oxygen, deoxyribose, and then a phosphate group, okay? So let's the big deals here. So obviously Watson and Crick, everyone loves them. 1953, they released their paper in Nature magazine about how meow, 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 this is double helix. Great. So, but this was not just kind of came out of nowhere. They also were really young guys. They were they were closer to your age than my age. They were like in their early to mid 20s. So feel bad about yourself for what you've done with your life when they were like 23 discovering DNA, or uncovering DNA, I should say. So this all started, I'm going to say this because I want to, and because a lot of what we know about science happens, people who get credited with the discovery or when the publishing, when they make this information public, we give them the credit. But so much had happened before that. And one in particular was Rosalind Franklin. And she was a woman, and she was credited with taking the photo, which is right here, that led to Watson and Crick then saying, oh, wait a minute, this is what we think DNA is. Now, she was a lady. It was the 1940s, 1950s, and her job, not only did she take this photo, but she also recognized, wait a minute, I think this is what DNA, I think I have a picture of the structure of DNA. So it was not only Rosalind Franklin, it was also Maurice Wilkins, but she was Watson and Crick's main lab assistant. No big deal, because she worked in X-rays, right, and crystallography, which is a type of X-ray. She spent her life's work in around radiation, died of 36, no big deal. So this is kind of what kind of was the one that this picture really allowed Watson and Crick to say, hey, yes, we have this paper, we have this presentation coming up, we're gonna come out and say that this is what we think DNA is. We did. So here we are, two party guys. Actually, there's stories. These guys are weird. I think, I should know this. I think Francis Crick became like a Holocaust denier. I shouldn't say that, but one of them had some very, very radical political views. When I was in college at Marquette, one of my instructors apparently had a romantic relationship with James Watson, and she had like a face like a boot. So who knows? Things were weird back then. Yeah. So these two, Watson and Crick, are the ones that came out and said, yes, this is what DNA looks like, and this changed everything. Changed our understanding of what DNA is, what DNA does, what DNA can do. So just some basics on what the structure of DNA. You have that phosphate backbone, right? So this is your phosphorus. This phosphorus, that line is that backbone part. It's right here. You also have this sugar part that's connected to the phosphates. Right, so phosphates here, and the sugar parts are here. And then you have your base pairing. So your base pairings are going to be adenine, guanine, cytosine, and thymine, or thymine, I should say. So that's here. These are held together by hydrogen bonds. okay? So they're not necessarily weak bonds, or, I'm sorry, they're not necessarily strong bonds, but they're protected by the sugar phosphate backbone here. So they don't necessarily have to be that strong because this is keeping them protected. Now, the direction of DNA. This 5' and this 3' has to do with the location of carbon atoms, okay? So when we talk about something being 5', we're referring to a carbon atom in a specific location. Same with 3'. Do you have to know all that? Not necessarily, but I do need you to know that we're going in reverse directions. So basically you've got, in this side, you've got your ribose looking like this. On the 3', your ribose is upside down. That's basically all this means, okay? And they run in opposition to each other. This allows for these guys to bond together. That's why that's critical. All right, DNA replication. We call it semi-conservative because you're starting with a parent molecule, okay? This is the OG right here, the OG of the DNA. And there's part of that OG stays. Part of the original molecule stays. That's why we call it semi-conservative. Total conservative would be you'd have nothing. You'd have this parent molecule you start with the parent molecule, and you end with the parent molecule. And you don't. You end up with part of it. So that's why it's semi-conservative. All right. So it takes enzymes on enzymes on enzymes to get this happen. It takes a village to get DNA to replicate. It takes a village of enzymes to get DNA to replicate. These are the three we're going to focus on: helicase, polymerase, and ligase. How do we know they're enzymes? Oh, great question. Remember, because they end in ASE. So again, you have this parent molecule, and they call it the daughter. I don't know why they call it the daughter, not the son. But you have this new strand of DNA, which is then linked and twisted in that double helix. So the helicase comes on in, like I said, Miley Cyrus on a wrecking ball, right? Comes on in and breaks those hydrogen bonds. They're very weak bonds. That's not too difficult to do. But as we know, enzymes are specific. So only helicase is the only one that can come on in and unwind the DNA strands. Then you've got polymerase. So what happens is you've got your helicase here, and you've got your polymerase adds. So it starts to add segments of DNA. Now, it starts at the 3' end. So what that means is it starts with the carbon at the 3' location. That's where it starts, okay? So it comes in and it adds, it starts to add essentially DNA fragments, which we know are a sugar, a phosphate, and a nucleotide, okay? These DNA fragments are basically kind of floating around in the nucleus. And the polymerase kind of comes in, grabs them, and puts them all together. Okay, that might have erased. If it did, I'll just continue. If it didn't erase, you have like some bonus audio. Your leading strand is like your firstborn, right? It's your firstborn child. It's where the polymerase first attaches, okay? It's the one that gets all the attention. There's baby photos upon baby photos of that firstborn. Same with that leading strand. It's where it starts, right? That polymerase is really trying with that leading strand. And it's where the DNA begins. The replication part, I shouldn't say begins, but it's where that new strand begins. It's when it's first attached. Again, in case I erased it when I said it, it also starts at the 3' end. Your lagging strand is going to be like your last born, right? You're trying. I mean, I think about, I'm the oldest of 3, and my parents were like really gunning hard with me. I had rules, I had curfew, I wasn't allowed to drink sugary drinks. My brother Nick comes along, he's eating Pop-Tarts for breakfast, lunch, and dinner, right? So your lagging strand, yeah, it's gonna work, stuff's gonna happen, but it lags behind because the leading strand is the first to get the daughter strand first to get replicated. The lagging strand is going to be the second part, so it lags behind. Because of that, you're going to see some gaps. We call those gaps Ozaki fragments, okay? So while this is continuous, it happens continuously. This part, the leading strand, is constantly being replicated. The lagging strand begins and stops, begins and stops, begins and stops, because it's kind of waiting on that leading strand to go first. Because it kind of comes in and comes out, there's gaps, and so there's big gaps in between the new DNA strands. That's where DNA ligase comes in. DNA ligase comes and fixes and connects those Ozaki fragments. That's the DNA ligase's job. Again, the leading strand is always kind of growing just as quickly as the replication fork opens up. So the minute the helicase comes in, the polymerase comes in, that leading strand there's replication going on there. The lagging strand has to kind of wait for that leading strand to go first. That's why we call it lagging. It has to wait for the leading strand to go. So, and also because you've got discontinuous. For the lagging strand, it's going to start, replication will start, then it'll stop, then it'll start, then it'll stop. And it makes more time, it takes more time for it to complete the daughter strand because it has to wait for the DNA polymerase to restart its work over and over again. Okay, the DNA polymerase can do just so much. So it's working real hard on that leading strand. It'll get to that lagging strand. You know, my youngest brother, he'll eventually get a bath. He'll eventually get his diaper changed. But mom's got other stuff going on, you know? So it has to kind of wait for that DNA polymerase to kind of get to it. So that's why it kind of lags behind, and also that's why you've got gaps. That's why you've got these gaps in the fragment, which we call the Ozaki fragments. So, big picture, how does this happen? It happens throughout the DNA, right? You have what's called replication bubbles. So it begins at a number of places. It doesn't always begin at one end or the other. It can happen in the middle of DNA. So replication can kind of start whenever it's time. Whenever, you know, we're right before mitosis, let's do this, let's replicate. What signals that replication are different proteins, different DNA in the cell that says, okay, it's time to go. Different enzymes that kind of trigger this DNA replication process. So what happens is, this is kind of what it looks like in a bigger picture. You've got the helicase, you've got the bubble, and the helicase starts, and it's going essentially in opposite directions. So you have this happening, and then you have the helicase, and then you have the polymerase that comes and forms the new strand, and you have this happening, boom, 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 boom. But it's not like unzipping necessarily. It's happening in two different, it's not like a zipper where one end opens along, you know, you open a zipper, you close a zipper, and it's along the line. Replication happens in these little bubbles throughout the DNA strand. Moves in both directions until you have this. So keep in mind, for every replication bubble, which is this, there's going to be two forks. This is fork number 1. This is fork number 2. So this is just, for example, right here, this is just one fork. This is also, this same thing is happening kind of opposite over here. Okay? And again, you have the 3' you know is going to be the leading strand, the one where DNA polymerase starts at, and then your lagging strand is going to be here. And your DNA polymerase will get to it, it'll get there, but it has to kind of start and stop because it's focused so much on that leading strand. And that's it. Woo! So my hope is that as I was kind of walking you through these slides, you were taking notes on the either like the slides I gave you, or you were just maybe just taking notes through your notebook. So right now what I'd like you guys to do is, just for clarification, you know, at the bottom of the slides or at the bottom of the piece of paper that you're working on or your notebook, give me a couple questions. If there's things that you need clarity on, if there's things you want to make sure that I explain in further detail, write that down so we can go over that first thing tomorrow. Okay, celebration, great.